Now we are going to study the unit 4, that is the plant physiology. 3 units have I already done it. It's on my channel. Now unit 4, plant physiology. So photosynthesis in higher plants. We see the description. We read it little bit. Okay, so I got something important in this paragraph, like the two perspectives essentially rested on two levels of organization of life forms and phenomena. One described at organismic and above level of organization, and while the second described at cellular and molecular level of organization. Now the first resulted in ecology, so the organismic level of organization, they resulted in ecology and related disciplines. The second resulted, that is the uh, described that cellular and molecular level of organization, resulted in physiology and biochemistry. Now this is done. This is Melvin Kelvin. Now what he does, just C14, he used the radioactive carbon dioxide and proposed that plants change light energy to chemical energy by transferring an electron in an organized area of pigment molecules and other substances. Okay, Kelvin and co-workers put radioactivity to beneficial use. So this is Melvin Kelvin. Now the photosynthesis in higher plants, chapter 11. The context are: what do we know? Early experiments. Where does photosynthesis takes place? How many pigments are involved in photosynthesis? What is light reaction? The electron transport. Where are the ATP and NADPH used? The C4 pathway, photorespiration, factors affecting photosynthesis. Now, what do we know? What we know? All animals, including human beings, depend on plants for their food. Right? The green plants make, or rather synthesize, the food they need through photosynthesis and are therefore called autotrophs. Okay. The autotrophic nutrition is found only in plants, and all other organisms that depend on the green plants for food are heterotrophs. So autotrophic nutrition is found only in plants. Green plants carry out photosynthesis, a physico-chemical process by which they use light energy to drive the synthesis of organic compounds. Ultimately, all living forms on Earth depend on sunlight for energy. The use of energy from sunlight by plants doing photosynthesis is the basis of life on Earth. Now what it says, like, the use of energy from sunlight by plants doing photosynthesis is the basis of life on Earth. Now, photosynthesis is important due to two reasons. It is the primary source of all food on Earth. It is also responsible for the release of oxygen into the atmosphere by green plants. So what do we know? Okay, some simple experiments you may have done in the earlier classes have shown that chlorophyll, that is green pigment of the leaf, light, CO2, are required for photosynthesis to occur. Okay, chlorophyll, that is green pigment of the leaf, light, CO2 are required for the photosynthesis to occur, right? Now, star formation in two leaves. A variegated leaf, or a leaf that was partially covered with black paper exposed to light. On testing these leaves for the presence of starch, it was clear that photosynthesis occurs only in the green parts of the leaves in the presence of light. Okay, another experiment. Okay, They found that the exposed part of the leaf tested positive for starch while the portion that was in the tube tested negative. This showed that CO2 was required for the photosynthesis. So KOH soaked cotton that absorbs CO2 inside the tube that show negative uh, that show negative results. This showed that CO2 was required for photosynthesis, right? Can you explain how this conclusion could be drawn? Early experiments. Now early experiment Joseph Priestley in 1770 performed a series of experiments that revealed the essential role of air in the growth of green plants. Priestley discovered oxygen in 1774. Priestley observed that a candle burning in a closed space, a bell jar, soon gets extinguished. Similarly, a mouse would soon suffocate in a closed space. He concluded that a burning candle or an animal that breathed the air both somehow damage the air. So burning candle and an animal, that is mouse, that breathe the air both somehow damage the air. But when he placed a mint plant in the same bell jar, he found that the mouse stayed alive and the candle continued to burn. Priestley's hypothesis as follows: plants restore to the air whatever breathing animals and burning candles remove. Now Jan Ingenhouse showed that sunlight is essential for the plant process that somehow purifies the air fouled by burning candles or breathing animals. Now Ingenhouse, in an elegant experiment with an aquatic plant showed that in bright sunlight small bubbles were formed around the green parts while in dark they did not. Later he identified these bubbles to be of oxygen. Hence he showed that it is only the green part of the plants that could release oxygen. I repeat it again now. Ingenhouse, in an elegant experiment with an aquatic plant showed that in bright sunlight small bubbles were formed around the green parts while in the dark they did not. Later he identified these bubbles to be of oxygen. Hence he showed that it is the only green part of the plants that could release oxygen. So this is what Jan Ingenhouse did, and he said that sunlight is essential for the plant process. Also the green parts that could release the oxygen. Now 1854, Julian von Sachs, for production of glucose when plants grow. Glucose is usually stored as starch, right? That the green substance in plants, chlorophyll, is located in special bodies and later called chloroplast within the plant cells. He found that green parts in plants is where glucose is made and that the glucose is usually stored as starch. So Julian von Sachs in 1854, he told like glucose is usually stored as starch. Now consider the interesting experiments done by T.W. Engelmann. Okay, he used a prism. He split the light into its spectral components and then illuminated a green alga, that is cladophora is a green alga, okay, placed in suspension of aerobic bacteria. Now the bacteria were used to detect the sites of O2 evolution. He observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. A first action spectrum of photosynthesis was thus described, right? It resembles roughly the absorption spectra of chlorophyll A and B, but this represents roughly the absorption spectra of chlorophyll A and B, right? Okay, that plants could use light energy to make carbohydrates from CO2 and oxygen, CO2 and water. Okay, CO2 plus water in the presence of light, it forms the carbohydrate, that is CH2O plus O2, right? CO2 plus H2O in presence of light, it forms the carbohydrate, that is CH2O plus O2, where CH2O represented a carbohydrate, that is glucose, a 6-carbon sugar. Okay, now Cornelius von Van Niel, okay, he studied on purple and green bacteria, demonstrated that photosynthesis is essentially a light-dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrates, thus is expressed as H2O to H2A plus CO2. So hydrogen from oxidizable compound, it reduces CO2 into CH2O carbohydrate, and it gets itself H2A gets itself what oxidize. Okay, and water response. So 2HTA plus CO2 light 2A CH2O plus H2O. Now in green plants, H2O is the hydrogen donor in green plants and is oxidized to oxygen, as I said. In so H2 is oxidized to oxygen. Now some organisms do not release CO2 or O2 during photosynthesis, when H2O instead is the hydrogen donor for purple and green sulfur bacteria. The oxidation product is sulfur or sulfate depending on the organism and not O2. Okay, hence he inferred that the O2 evolved by the green plant comes from H2O, not from the carbon dioxide. So this was later proved by using radio isotope techniques. Okay. So 6CO2 plus 12H2O in presence of light, it forms C6H12O6 plus 6H2O plus 6O2. So this is the reaction, like where 6C6H12O6 represent glucose. The O2 release is from water. This was proved using radioisotope technique. Note that this is not a single reaction but description of a multi-step process called photosynthesis. Can you explain why 12 molecules of water as substrate are used in the reaction given below? So 12 H2O molecules, because the electrons needed during the transfer of electron from PS2 to PS1, all that things. Okay, so where does photosynthesis takes place? Now this is the paragraph we are going to study. So where does photosynthesis takes place? The green leaf or in the chloroplast, right? Can you name some other parts where you think photosynthesis may occur? But photosynthesis does takes place in the green leaves of plants, but it does so also in other green parts of the plants. That the mesophyll cells in leaves have a large number of chloroplasts. Usually the chloroplasts align themselves along the walls of the mesophyll cells such that they get the optimum quantity of the incident light. When do you think the chloroplast will be aligned with their flat surfaces parallel to the walls? When would they be perpendicular to the incident light? Okay. Within the chloroplast there is membranous system consisting of grana, the stroma, lamella, and the matrix stroma. There is a clear-cut division of labor within the chloroplast. The membrane system is responsible for trapping the light energy and also for the synthesis of ATP and NADPH. In stroma, enzymatic reactions synthesize sugar, which in turn forms starch. Okay, the former set of reactions, since they are directly light driven, are called light reactions. Okay, the former set of reactions, since they are directly light driven, are called light reactions, right? That is photochemical reactions. The latter are not directly light driven but are dependent on the products of light reactions, that is ATP and NADPH. Hence, to distinguish the latter, they are called by convention as dark reactions, carbon reactions. However, this should not be constructed to mean that they occur in darkness or that they are not light dependent. Yes, they are dependent on the light, but they are indirectly dependent on the light. So this is the chloroplast, and it has an outer membrane, then there is an inner membrane, then there is a stroma lamella, then there is a grana, and then there is a stroma, then there are ribosomes, then there are starch granules, then there are lipid droplet. So how many types of pigments are involved in photosynthesis? Like chlorophyll A, chlorophyll B, xanthophylls, and carotenoids. So like chlorophyll A, like bright or blue green in the chromatogram. In chlorophyll B, yellow green, xanthophylls yellow, and carotenoids yellow to yellow orange. Okay, see. So chlorophyll A, bright or blue green. Remember this color, very important. Chlorophyll A is bright or blue green, chlorophyll B yellow green, xanthophylls yellow, and carotenoids yellow to yellow orange. So these are like pigments are substances that have an ability to absorb light at specific wavelengths. Okay, the ability of chlorophyll A pigment to absorb lights of different wavelengths. The spectrum is Vibgyor. Can you determine the wavelength at which chlorophyll A shows the maximum absorption? Does it show another absorption peak at any other wavelengths too? If yes, which one? Showing the wavelengths at which maximum photosynthesis occurs in a plant. There is maximum absorption by chlorophyll A, that is in the blue and red regions, also shows higher rate of photosynthesis. Hence, we can conclude that chlorophyll A is a chief pigment associated with photosynthesis. Okay, so can you see that there is a complete one-to-one overlap between the absorption spectrum of chlorophyll A and the action spectrum of photosynthesis? Yes, there is. As you can see, the first diagram shows like absorbance the absorbance of light by chloroplast pigments, that is you can see chlorophyll A, then chlorophyll B, then chlorophyll, then carotenoids, as you can see. Then there is rate of photosynthesis measured by CO2 released. So this is the second diagram, that is rate of photosynthesis, that is action spectrum of photosynthesis, right? And then there is the third diagram shows the light absorption. As you can see, wavelength of light in nanometers, rate of photosynthesis, and then absorptions. They just overlap one to one another, like so. A represents what? A represents showing the absorption spectrum of chlorophyll A, B, and the carotenoids. A, B, and carotenoids. A, B, and carotenoids. Then graph showing action spectrum of photosynthesis, the second graph, and graph showing action spectrum of photosynthesis superimposed on absorption spectrum of chlorophyll A. So this is it. Now coming to the next paragraph. So these graphs together show that most of the photosynthesis takes place in the blue and red regions of the spectrum. Some photosynthesis just takes place at the other wavelengths of the visible spectrum too. Okay, though chlorophyll is the major pigment responsible for trapping light, other thylakoid pigments like chlorophyll B, so these are the thylakoid pigments like chlorophyll B, xanthophylls, and carotenoids, which are called accessory pigments, also absorb light and transfer the energy to chlorophyll A. Indeed, they not only enable a wide range of wavelength of incoming light to be utilized for photosynthesis, but also protect chlorophyll A from photooxidation. So what is light reaction? Here, so light reaction or the photochemical phase include light absorption, water splitting, oxygen release, and the formation of high energy chemical intermediates like ATP and NADPH. Like several protein complexes are involved in the process. The pigments are organized into two discrete photochemical light harvesting complexes, LHC, within the photosystem 1, PS1, and photosystem 2, PS2. So these are named in the sequence of their discovery, not in the sequence in which they function during the light reaction. The LHC are made of hundreds of pigment molecules bound to proteins. Each photosynthesis has all pigments except one molecule of chlorophyll A, forming a light harvesting system also called antenna. Has all the pigments except one molecule except one molecule of chlorophyll A, forming a light harvesting system also called antenna. These pigments helped these pigments help to make photosynthesis. These pigments help to make photosynthesis more efficient by absorbing different wavelengths of light. The single chlorophyll A molecule forms the reaction center. The reaction center is different in both the photosystems, right? In PS1, the reaction center chlorophyll A has an absorption peak at 700 nanometer, hence is called P700, while in PS2 it has an absorption maximum at 680 nanometer and is called the P680. Okay, so this is it. Now These are the pigment molecules, they are bound to each other. The photon falls on it at the reaction center, and then there is a primary acceptor. So okay, now the electron transport. In photosystem 2, the reaction center chlorophyll A absorbs 680 nanometer wavelength of red light, causing electrons to become excited and jump into an orbit farther from the atomic nucleus. Now these electrons are picked up by an electron acceptor, which passes them to an electron transport system consisting of cytochromes. This movement of electrons is downhill in terms of an oxidation reduction or redox potential scale. Now as you can see, the electrons are not used up as they pass through the electron transport chain, but are passed on to the pigments of the photosystem 1. Okay, so the electrons released in photosystem 1, they are not used up, but they are passed to from photosystem 2 to photosystem 1, right? Simultaneously, the electrons in the reaction center of PS1 are also excited when they receive a red light of wavelength 700 nanometer and transport to another acceptor molecule that has a greater redox potential. These electrons are then moved downhill again, this time to a molecule of energy rich NAD P+. The addition of these electrons reduces NAD P+ to NAD PH plus H+. This whole scheme of transfer of electrons starting from the uphill to the acceptor, down the electron transport chain where the ATP is formation, and then to PS1, and then excitation of electrons, then transport to another acceptor, and finally downhill to NAD P+, reducing it to NAD PH plus H+. Now this is called the Z scheme due to its characteristic step. Okay, now splitting of water. As you can see this diagram, scheme of light reaction. Light is falling on the LHC, then H2O gets converted into 2 electron plus H2O plus H plus oxygen. As you can see, this is acceptor molecule, then photosystem 2, and then electron transport system, then LHC, then light harvesting complex, then electron acceptor, then photosystem 1, then NADP plus is converted into NADPH, so plus H plus. So this is the Z scheme. Now the splitting of water. How does PS2 supply electrons continuously? The electrons that were moved from photosystem 2 must be replaced. So this electron must be replaced. This is achieved by electrons available due to splitting of water. The splitting of water is associated with the PS2. Water is split into 2H plus oxygen atom plus electrons. This creates oxygen, one of the net products of photosynthesis. The electrons need to replace those removed from the photosystem 1. Photosystem 1 are provided by photosystem 2. So this 2 H2O to 4H plus O2 plus 4 electrons. So we need to emphasize here that the water splitting complex is associated with the PS2, which itself is physically located on the inner side of the membrane of the thylakoid. Okay, then where are the protons and oxygen form likely to be released? In the lumen or on the outer side of the membrane. Okay, now the cyclic and non-cyclic photophosphorylation. So okay, the living organisms have the capability of extracting energy from oxidizable substances and store this in the form of bond energy. Special substances like ATP carry this energy in their chemical bonds, process through which ATP is synthesized by cells in mitochondria, and chloroplast is named phosphorylation. Okay, now the photophosphorylation is the synthesis of ATP from ADP plus inorganic phosphine in the presence of light. When the two photosystems work in a series, first PS2 and then PS1, a process called non-cyclic photophosphorylation occurs. So first is the PS2 and then one is the PS1, and then it is a non-cyclic photophosphorylation occurs, right? The two photosystems are connected through an electron transport chain as seen earlier in the Z scheme. Both ATP and NADPH plus H plus are synthesized by this kind of electron flow. Okay, so as you can see, cyclic photophosphorylation, light is followed and chlorophyll P700, then it acceptor, then photosystem 1, then electron transport system, it continues. Okay, so this is of cyclic photophosphorylation. So when only PS1 is functional, the electron is circulated within the photosystem, and the phosphorylation occurs due to the cyclic flow of electrons. A possible location where this could be happening in the stroma lamella. Okay, while the membrane or lamella of the grana have both PS1 and PS2 In the stroma lamella, membranes lack PS2 and as well as NADP reductase enzyme, and this is why they show only cyclic photophosphorylation in the stroma lamella, not on the thylakoid membrane or lamellae of the grana. The excited electrons does not pass on to NADP plus but is cycled back to the PS1 complex through the electron transport chain. The cyclic flow hence results only in the synthesis of ATP but not of the NADPH plus H plus. Cyclic phosphorylation also occurs when only light of wavelengths beyond 680 nanometer are available for excitation. I repeat it again. Cyclic photophosphorylation also occurs when only light of wavelengths beyond 680 nanometer are available for excitation. Now chemoosmotic hypothesis. So this is the chemoosmotic hypothesis. Okay, chemoosmosis requires a membrane, a proton pump, a proton gradient, ATP synthase. Chemoosmosis requires a membrane, that is thylakoid membrane, a proton pump to send this hydrogen H plus ion back into the lumen. Okay, to restore the gradient, proton gradient, right? And then there is a proton gradient and ATP synthesis. ATP synthesis, you can see CF naught embedded in the thylakoid membrane, then on the outer side there is a CF1, okay, that has the ATP synthesis. Okay, so this is all. Like now so along with the NADPH produced by the movement of electrons, the ATP will be used immediately in the along with the NADPH produced by the movement of electrons, like that in the stroma side. The ATP will be used immediately in the biosynthetic reaction taking place in the stroma responsible for fixing CO2. So ATP and NADPH will be used directly, right, in the stroma responsible for fixing CO2 and synthesis of sugars, right? Okay, this gradient is important because it is the breakdown of this gradient that leads to the synthesis of ATP. Okay, that leads to the synthesis of ATP. The gradient is broken down due to the movement of proton across the membrane to the stroma through the transmembrane channel of the CF node of the ATP synthase. Transmembrane channel of the CF node of the ATP synthase. So ATP synthase has the transmembrane channel, that is CF node, and it has the CF1 as well on the outer side. Okay, so the ATP synthase enzyme consists of two parts. One is called the CF node. Okay, so ATP enzyme has the two parts. As one is called the CF node, is embedded in the thylakoid membrane and forms a transmembrane channel that carries out facilitated diffusion of protons across the membrane. The other portion is called the CF1 and protrudes on the outer surface of the thylakoid membrane on the side that faces the stroma. Okay, now the breakdown of the gradient provides enough energy to cause a conformational change in the CF1 particle of the ATP synthase, which makes the enzyme synthesis several molecules of energy packed ATP. Okay, now the three reasons how this proton gradient build up. Since splitting of the water molecule takes place on the inner side of the membrane, the protons or hydrogen ions that are produced by the splitting of water accumulate within the lumen of the thylakoids. So this is the one reason. As you can see, H2O, that is oxidation of water to O2 plus H plus. Now another reason is, as electrons move through the photosystems, protons are transported across the membrane. This happens because the primary acceptor of electron, which is located towards the outer side of the membrane, transfers its electron not to an electron carrier but to an hydrogen carrier. So electron uses an hydrogen carrier instead of using the electron carrier. Hence, from the stroma side, there is an hydrogen carrier which is being used in the electron transport. So the H plus is getting reduced. The concentration is getting decreased on the stroma side. So hence this molecule removes a proton from the stroma while transporting the electron. when this molecule passes on its electron to the electron carrier on the inner side of the membrane, the proton is released into the inner side or the lumen of the, so outside H plus gets transferred to the inside lumen. Okay, now third is the NADP reductase enzyme is located on the stroma side of the membrane. Okay, along with electrons that come from the acceptor of electrons of PS2, protons are necessary for the reduction of NADP plus to NADPH plus H plus. These protons are also removed from the stroma. Now these protons are also removed from the stoma. Okay, hence within the chloroplast, protons in the stroma decrease in the number, while in the lumen there is an accumulation of protons. This creates a proton gradient across the thylakoid membrane as well as a measurable decrease in the pH in the lumen. Okay, now what are the ATP and NADPH used? As you can see all, and I'm going to explain this diagram a little bit more. You see, okay, in here I left something. Okay, this paragraph is about chemosynthesis. So chemo-osmotic hypothesis is the synthesis of ATP. Now ATP synthesis is linked to the development of a proton gradient across a membrane, right? This time these are the membranes of thylakoids. So in the chloroplast, these are the membranes of the thylakoids, right? There is one difference though. Here the proton accumulation is towards the inside of the membrane, that is in the lumen. Okay, in respiration, protons accumulate in the intermembrane space of the mitochondria. So mitochondria has two membranes, so in between the two membranes the protons accumulated in their intermembrane space of the mitochondria when electrons move through ETS, like. So this is it. So this is the diagram, like you see, stroma, low H plus, light falls on PS2 P680 nanometer. Okay, then electron splitting of water, then electron to the plastoquinone, like, then taking the H plus carrier from the stroma side to the cytochrome B6F complex. Now it releases H plus inside the lumen. Now it giving to the plastocyanin acceptor, then it is giving to the P700, that is PS1, and this light falls on it, then FNR, then NADP plus H plus, it gets converted into NADPH. Okay, NADPH, right? So high electrochemical potential gradient in here. Just remember the path, like PS2, then plastoquinone, then cytochrome B6, then plastocyanin, and then PS1, right? So this is it. Now where are the ATP and NADPH used? Okay, so we learned that the products of light reactions are ATP, NADPH, and O2, right? These are the products of light reactions, right? Of these, oxygen diffuses out of the chloroplast, while ATP and NADPH are used to drive the process leading to the synthesis of food, more accurately sugars. This is the biosynthetic phase of the photosynthesis. This process does not directly depend on the presence of light, but is dependent on the products of the light reaction, that is ATP and NADPH, besides CO2 and H2O. You may wonder how this could be verified. It is simply immediately after light becomes unavailable, the biosynthetic process continues for some time and then stops. If then light is made available, the synthesis starts again. So this is one. So we calling the biosynthetic phase as the direct reaction is a misnomer. Okay, so ATP and NADPH are used in the biosynthetic phase, right? O2 is combined with H2O to produce H2N or sugars. Okay, the first product formed when CO2 is taken into a reaction or fixed during the World War II. Okay, with using the radioisotopes to beneficial use, Melvin Kelvin, radioactive C14. In the algal photosynthesis study led to the discovery that the first CO2 fixation product was 3-carbon organic acid. Now he called it Kelvin cycle. The first product was 3-phosphoglyceric acid or PGA. How many carbon does it have? It has 3 carbon, right? It has 3 carbon but 2 molecules. Now PGA as the first product of CO2 fixation. All plants have a PGA as the first CO2 as the first product of CO2 fixation, or whether any product was formed in another plants. So the first stable product of CO2 fixation was again an organic compound, but one which had 4 carbon atoms in it. So this time it was like in C3 it was 3 carbon first product, but now it has 4 carbon atoms in it. So this acid was identified to be oxaloacetic acid or OAA. Since then CO2 assimilation during photosynthesis was said to be of two main types: those plants in which the first product of CO2 fixation is a C3 acid, that is PGA, that is the C3 pathway, and those in which the first product was a C4 acid, that is OA, oxaloacetic acid, that is the C4 plants. These two groups of plants showed other associated characters that will be discussed later. Now the primary acceptor of CO2, as we can see, there is a dark reaction. How many carbon atoms would a molecule have which after accepting CO2 would have 3 carbons PGA? Okay, the acceptor molecule was a 5 carbon ketose sugar, 5 carbon ketose sugar, ribulose bisphosphate, RUBP, like ribulose bisphosphate, right? Believe that the first product was a C3 acid. The primary acceptor would be a 2 carbon compound. They spent many years, but it was found that it was the 5 carbon RUBP, the Kelvin cycle now. So all photosynthetic plants, it does not matter whether they have C3 or C4 pathways. So Kelvin pathway operates and where the sugar is synthesized, and the RUBP was also generated. Kelvin cycle operates in a cyclic manner. Sugar is synthesized, right? Under now the Kelvin cycle occurs under 3 stages, like carboxylation, reduction, and regeneration. Now carboxylation. So in carboxylation is the fixation of CO2 into a stable organic intermediate. Carboxylation is the most crucial step of the Kelvin cycle, where CO2 is utilized for the carboxylation of RUBP. This reaction is catalyzed by an enzyme RUBP carboxylase, which results in the formation of 2 molecules of 3 PGA, the 3-phosphoglyceric acid. Since this enzyme also has an oxygenation activity, it would be more correct to call it RUBP carboxylase. We call it RUBP carboxylase or oxygenase or Rubisco. You see, so this is the carboxylation. It is the first step, and the second step is the reduction. These are the series of reactions that lead to the formation of glucose. The steps involve utilization of 2 molecules of ATP for phosphorylation and 2 of NADPH for reduction per CO2 molecule fix. So I repeat it again. There are a series of reactions that lead to the formation of glucose. The steps involve utilization of 2 molecules of ATP for phosphorylation and 2 of NADPH for reduction per CO2 molecule fix. The fixation of 6 molecules of CO2 and 6 turns of the cycle are required for the formation of 1 molecule of glucose from this pathway. So this is the reduction. Now regeneration. Regeneration of the CO2 acceptor molecule RUBP is crucial if the cycle is to be continue uninterrupted. The regeneration step requires 1 ATP for phosphorylation to form RUBP. Okay, so carboxylation is the first step, then reduction, and then regeneration. Now you can see here. Hence, for every CO2 molecule entering the Kelvin cycle, 3 molecules of ATP and 2 of NADPH are required, right? 3 of ATP and 2 of NADPH required. Why? Because in the regeneration there is 1 ATP is used, and in reduction there is 2 ATP is used and 2 NADPH is used. So total is 3 ATP and 2 NADPH are used in overall the 1 turn of 1 Kelvin cycle, right? It is probable to meet this difference in number of ATP and NADPH used in the direct reaction that the cyclic process. You see the extra cyclic process runs, so that is for the extra ATP. Now this is getting cleared why this happens. To make 1 molecule of glucose, 6 tons of the cycle are required. 1 ton one CO2 is fixed, but we required in glucose 6 carbon atoms, so we need 6 tons of the cycle to be required. Work out how many ATP and NADPH molecules will be required to make one molecule of glucose through the Kelvin pathway. It might help you to understand all of this if we look at what goes in and what comes out of the Kelvin cycle. In, so 6 CO2 in and out, it gives 1 glucose. If 18 ATP, it gives 80 ADP adenosine, okay. Then 12 NADPH, it gives 12 NADP. So NADPH, it gives 12 NADP. Now I'm explaining this cycle a little bit. Now the Kelvin cycle proceeds in 3 stages: carboxylation, during which CO2 combines with ribulose 1,5-bisphosphate; carboxylation, during which CO2 combines with ribulose 1,5-bisphosphate. Now reduction, during which carbohydrate is formed at the expense of the photochemically made ATP and NADPH. Now regeneration, during which the CO2 acceptor ribulose 1,5-bisphosphate is formed again, so that the cycle continues. Okay, I read it again. The Kelvin cycle proceeds in 3 stages: the carboxylation, during which CO2 combines with ribulose 1,5-bisphosphate, during which CO2 combines with ribulose 1,5-bisphosphate; reduction, during which carbohydrate is formed at the expense of the photochemically made ATP and NADPH; regeneration, during which the CO2 acceptor ribulose 1,5-bisphosphate is formed again, so that the cycle continues. Okay, so this is it. You can see first step is the carboxylation, that is CO2 plus H2O, ribulose 1,5-bisphosphate. Then there is 3-phosphoglycerate formation, and then there is the reduction, that is ATP and NADPH. Then there is ATP and NADPH, that is the reduction, and ADP plus inorganic phosphate, and then triose phosphate, that is sucrose, and then there is starch. then triose phosphate, that is formation of sucrose and then starch, and then there is the regeneration, that is ATP gets converted into, okay. So this is it, like regeneration, ATP is being used. So this is all right now. Okay, so carboxylation, then reduction, and then regeneration. So this is all the goes, like this is the Kelvin cycle. Now the next will be like next will be like C4 pathway and all that.